Hello everyone, welcome to the second lecture of MSc 101 course. In this lecture, I will mainly talk about jam theory and Koch postulates, and these are very important. In relation with the disease. Okay, but before talking about them, I will talk about the fermentation process. Basically, I will try to give you a brief idea about fermentation process. So, what fermentation process is? We microbiologists we usually refer fermentation as the process by which carbohydrates is converted into alcohols and carbon dioxide or organic acids. Okay, this is a common definition. Change of carbohydrates into alcohols with the help of microorganisms. Usually, we microbiologists we like to give this definition, but the fermentation by the word fermentation, its meaning is broader. So, usually, the by fermentation means change of a substrate into another. For example, change of carbohydrates into alcohol. Okay, so change of a substrate into another substrate by using biological molecules. So here the biological things can be microorganisms, cell, animal cell or plant cell. It also can be enzyme. Okay, so that means we can thus we can define fermentation as change of a substrate into a simpler form with the help of biological molecules. Okay, so anyway, in case of fermentation where we use microorganisms, many different microorganisms are used. Bacteria are used, yeast are used. Okay, for example, to produce alcohol using fermentation process in industry, yeasts and bacteria are used. Okay, and this process happen under anaerobic condition. Okay, anyway, I want to give you another information. So, if you search for the definition of fermentation in the internet, okay, sometimes you will get that fermentation is a anaerobic process, or sometimes you will get that fermentation is a aerobic process. So, which one is correct? Well, both are correct. Okay, fermentation can be an anaerobic process. Fermentation can be an aerobic process. So, let me explain a little bit. So, this process, like suppose you want to produce alcohol, so what we will do, you need to convert carbohydrates into alcohol. This change will be, this change will occur in anaerobic condition. Okay, suppose say yeast will change carbohydrate into alcohol in the absence of oxygen. That means in anaerobic condition. Okay, how about let's imagine about another scenarios. So you all know about the baking yeast, right? To make bread, yeasts are used. So that means, uh, suppose you want to sell yeast. What you have to do? You have to produce lots of yeast, and using fermentation process. Okay, so in that case, actually you need aerobic condition because to grow yeast itself need oxygen. That means aerobic conditions. Okay, so thus we now understand that fermentation can be both aerobic and anaerobic. Okay, so now let's talk about little bit about the history of fermentation. So from where this idea came. Okay, in old times, okay, when there was no very fast ship vessels like nowadays. Okay, in old days the ships were not that speedy, so that's why it used to take very long time to ship goods or things from one country to another, several months. So at that time, several French merchants they observed one thing. They found that whenever they transport their alcoholic beverages, so for example beer from one country to another, sometimes those beer used to get spoiled. Okay, the taste become very sour taste means acidic taste. Okay, so actually at that time people didn't have any idea that microorganisms can do that. So those French merchants they asked a very famous scientist Pasteur, Louis Pasteur. They asked Louis Pasteur to solve this, to find out why this happened. Okay, you know, at that time many scientists used to believe that air convert the sugars in the in these fruits into alcohol. At that time people, many scientists used to believe that alcohol are produced from sugar by air. But later Pasteur actually did many experiments, and eventually he found that not the air, but microorganisms called yeast, they convert the sugars to alcohol in the absence of air. And there are some microorganisms which can further react with this ethanol, okay, and make acid such as ethanol acids and aldehydes, okay, such as ethanol from acetaldehyde, okay, and acrolein phenol. And as acids are produced, the taste of those ethanol turned into sour, sour, okay. So from that, I mean, from these experiments, people get the idea of fermentation. Okay, that means we can produce something. That means we can convert something using microorganisms. Okay. Well, so that's all for the fermentation. But don't get disheartened. I mean, it was very brief, but don't get disheartened. Actually, we have a separate course named fermentation technology. So in that course, you will mainly learn about fermentation in more details, much more details. Okay. Okay. So now let's talk about the germ theory and Koch postulate. So what is germ theory? Nowadays, we know that germ can cause disease, but I mean, for long period, okay, for centuries, people used to believe that disease are caused because of the sin, papel fossil, okay. So I mean, people used to believe that, I mean, we get diseased as a punishment for our sins, our bad character or poverty, okay. But then, I mean, eventually, Robert Koch, this scientist, I mean, he did many experiments, and he finally convinced the people. He was able to finally prove that, okay, the diseases are not caused by the sin. Okay, diseases are caused by germs. Okay, and this is actually called the germ theory. I mean, by the term germ theory of disease, we mean that germs cause the disease. Okay, and not only this one, Robert Koch did also did many series of tests, and he proved how germ cause disease. Basically, he established the link. Okay, the link between microbes, that means germs, with the infectious disease. Okay, so in doing this, he did series of experiments, and from that experiments, he he made some statements. These statements are called postulates, Koch postulates. Okay, so now we'll talk about Koch postulate. Before that, I want to share information with you about Robert Koch. I mean, Robert Koch not only did this. I mean, not only he established the germ theory and gave some postulates which create a link between the microbes and infectious disease. Also, he did tremendous contribution to science, especially in the from the field of microbiology. Okay, such as he identified causative agents of anthrax and tuberculosis. He developed several techniques. Okay, such as he developed the technique of use of agar as a solid medium. So what is medium? I'll talk about it in another lecture. Okay, and also Koch invented nutrient broth and nutrient agar. So nutrient broth and nutrient agar are actually food for the microbes. Okay, so if we want to cultivate or culture microbes, we need to give them some food. So usually we give them this food either in liquid form, which is called broth, or in solid form, which is called agar. Okay, I'll talk about this in more details in another lecture. Okay, so now talks about those statement of postulates. Okay, so before explaining these postulates, I would like to tell you why these postulates are important. Okay, so we know that germ cause disease. Okay, simple thing, but in reality, I mean, as a scientist, you just cannot say that this germ caused this disease. Suppose, for example, suddenly some people they started to get sick. Okay, and say you are a scientist and you did some experiments, and then finally you predicted that this germ can may cause this disease. Look at this word, what I said. This germ may cause this disease. You cannot say it in a very confident way. You cannot say it surely that this germ caused this disease. To say that, you actually have to you actually have to prove some statement. You have to prove it through the tests. So what are those tests and what you need to prove? Okay, so you need to prove the statements which were proposed by Koch. Okay, so if you want to say that this germ caused this disease, then experimentally you have to prove the Koch postulates. Okay, so now let's talk about the Koch postulates. So what are the postulates which were, what are the statements which were proposed by Koch? Okay, there. Koch proposed four statements. Okay, so rather explaining from here, I will explain from this slide. It has some pictures, so you need to understand. Okay, so Koch had proposed four postulates. Number one, the suspected pathogenic organism should be present in all cases of the disease and absent in healthy animals. Okay, so suppose you suspected that these microorganisms cause this disease. So in that case, that suspected organism should be present in every disease host. If it is a human disease, in every human, or if it is an animal disease, in every animal. Okay, and the healthy host should be free from that microorganisms. That means there should be no microorganisms. There should be no that suspected microorganisms in the host body. Okay, so this is the first Koch postulate. Disease person, every disease person will have that suspected or microorganisms, but the healthy person, they will not have the suspected microorganisms. Okay, so then uh, postulate, second postulate, the suspected organism should be grown in pure culture. Okay, so what that means? Say, a person, the every person have that particular microorganism. So you need to isolate these microorganisms from the body of that host, and you have to culture them. Culture means grow. You have to grow them outside the host body. Actually, we can culture bacteria in our laboratory. Okay, and to do that, we use a dish. Those are called petri dish. We put some solid food on that petri dish, and then we culture or grow the bacteria on it. Okay, so second postulate is you should be able to isolate the suspected organism and grow them in the petri dish, which is called pure culture. Okay, so this was the second postulate. Now the third postulate. Okay, so the third postulate say that cells from a pure culture of the suspected organism should cause disease in a healthy animal. In the second postulate, you have isolated the microorganism and cultured it. Okay, so now what you have to do, you have to take this bacterial cell and inject them into healthy animal. Okay, inject them into healthy animal. So now, if the healthy animal, so after you injected the isolated microorganism into the healthy animal, that healthy animal should get sick. Okay, that means disease should be developed into that healthy animal. Okay, so now the fourth postulate is the organism should be isolated and shown to be the same as the original. Okay, so what you did in the third postulate, you isolated the bacteria that you cultured, injected into the healthy host get diseased, then you again have to isolate the bacteria from that diseased host, and again you have to culture them, and you have to show that this bacteria is same like the original one. Okay, so if you can experimentally prove all these postulates, then finally you can say that particular this microorganism cause this disease. Okay, so Koch postulates is very important. Okay, Koch postulates, that's why it's very important. Although Koch postulates actually have some limitations. Koch postulates were proposed long ago. Okay, so at that time, the knowledge about science was little. So that's why, it's normal that, Koch postulate will not be very accurate. But, I mean, actually it's true. Koch postulate have some limitations. But don't worry, it has some limitations, and also people actually did some modification to the original Koch postulates. Okay, so although original Koch postulates is faulty, it has limitations, but his steps, the steps that he describes, are still regarded as the gold standard. Gold standard for what? If you want to link between a microbes and a disease, you must have to, all these postulates must have to be proved. Okay, so nowadays we don't use the original postulate, we use the modification one. So I will talk about the modified Koch postulates, but before that, we need to understand the limitations that the Koch postulates have. Because if we don't understand the limitations, we will not be able to understand the modifications. So let's talk about the limitations and what limitations Koch postulates have. So these are some of the limitations. Number one, the causative microorganism might not be present in every stage of the disease. For example, Vibrio cholerae might not be present at the time of extreme vomiting and diarrhea because the bacteria release toxin which cause the diarrhea even in the absence of an organism. Original Koch postulates say that the suspected causative microorganism should be present in every healthy host. But it's actually not true for every case. Sometimes it can happen that the microorganisms is not there, but still that person is sick. Okay, one example is given that is Vibrio cholerae. Okay, Vibrio cholerae cause cholera. Okay, actually Vibrio cholerae, it produce a toxin. Okay, and that toxin can cause the diarrhea and extreme vomiting. Okay, so it can happen that the person get infected by Vibrio cholerae, and that Vibrio cholerae, it produced the toxin and it left the host body. But as the toxin is still there, that toxin can cause the disease. Okay, so it's kind of contradictory with the Koch postulate. So this is one limitation. This kind of scenario we cannot explain by the original Koch postulate. Okay, so another limitations: many organisms or entities cannot be grown in pure culture, such as prions. Prions is an actually you can say it's kind of a it's not a microorganisms. It's a very tiny molecules. Okay, and it causes disease, which name is Creutzfeldt-Jakob disease. Okay, this example is more difficult. Actually, there are many infectious diseases which are caused by different microorganisms, and we cannot culture those microorganisms in vitro. We cannot grow those microorganisms outside the host body. Okay, this is very common in the field of microbiology. But if you want to say that these microbes or microorganism cause this disease, according to Koch's postulate, you should be able to isolate the microbes and culture them in vitro, culture them in the petri dish outside the body. Okay, so this is one limitation. And this is one limitation of the original Koch postulate. We cannot culture every microorganisms. Okay, so now another limitations: not all hosts exposed to an infectious agent will acquire the infection. Okay, because according to the original Koch postulate, if you isolate that suspected microorganism from the diseased person and then reinfect a healthy person with that microbes, that healthy person should be sick. That healthy person should get diseased. But in reality, the thing is, suppose there are 10 people and all of them get infected by the microorganisms, but all of them will not get the disease. Why? Because every person, they have some difference in their immunity. Okay, some people can be more immune, their immune system is more strong, or some people, they can be already vaccinated. So if they are already vaccinated or they are already infected by the same bacteria or microorganisms, it is possible that they already have a specific immunity against that particular microorganism. So if they get infected by the suspected agents, it's likely that they may not get disease, because right, they may not get disease because they can be, I mean, their natural immune system can be very strong, okay, or they can be pre-exposed or vaccinated, okay. So this is another limitations or a scenario which we cannot explain by the original Koch postulate. Okay, so actually Koch postulates also have some other limitations, such as it is ethically unacceptable to expose a host experimentally to prove pathogen disease relationships. Suppose, for example, you are starting with human disease. Okay, so you isolated the microorganism from one diseased human, and then, according to Koch postulate, you need to infect a healthy person, a healthy human with this microorganism. Okay. We cannot do that because it is unethical. Okay, it is not ethically accepted. Okay. Okay, so we understand that Koch postulate has many limitations, but I told you the steps described by the Koch are actually, they're kind of gold standard. So what the scientists did, they modified the original Koch postulate in a way that we can fit them with the modern knowledge. Okay, so these are some modification of the Koch postulates. Okay, basically, I mean, the new methods that is the revised version of Koch postulates was proposed by many different scientists. Okay, many different scientists actually they proposed many different modifications. So here these are kind of summary, okay, which was proposed by Frederick and Rellman. Anyway, you can say that these are the summary of the proposed modified postulate. Okay, so let's talk about this summary. Please try these modifications very, very clearly. Okay, these are very important for a microbiologist. Okay, it's very important for a microbiologist to understand the Koch postulate and their modification. Why this modification was necessary? Okay, so let's talk about the modification of the original Koch postulates. So this is the modified version. Okay, so number one. A nucleic acid sequence belonging to a putative pathogen should be present in most cases of an infectious disease. Okay, and microbial nucleic acid should be found preferentially in those organs or gross anatomic sites known to be diseased, not in those organs that lack pathology. Okay, so in original Koch postulate, it was stated that the causative agent should be present in all the diseased person. But in modified version, what we are saying, that it's not necessary that the whole microorganism should be present. Rather, the nucleic acid of that microorganisms, if the nucleic acid of that microorganism is present, that's okay. Okay. So it's not necessary the whole microorganism should be present. Only the presence of nucleic acid will be good enough. In addition, it's not necessary that the nucleic acid should be present in all the organs of our body. Okay, there are different type of disease, right? Sometimes say only our kidney is diseased, our heart is okay, kidney is diseased. Okay, our heart is diseased but kidney, pancreas may be normal. Okay, so that means in many cases not all our organs are affected. Okay, so in many cases only one organ or some of the organs are affected. So if we can isolate the nucleic acid of that suspected or putative pathogen from those diseased organs, that's okay. Okay, so the healthy organs, they should not contain the nucleic acid of a putative pathogen or the disease pathogen. Okay, okay. Now another modification. Fewer or no copies of pathogen associated nucleic acid sequences should occur in hosts or tissues without disease. I mean, according to original Koch postulates, healthy person should not contain any putative pathogens. Okay, but in reality, actually, what happens? Even the healthy person, they may sometimes contain few number of microorganisms. Okay, so in the modified Koch postulates, it's stated this way, that the healthy host can contain few few nucleic acids of that putative pathogen, or either they will contain no pathogen or no nucleic acid of the pathogen, or they may contain few nucleic acid of the pathogen. Okay. Okay, now another modification. With resolution of disease, the copy number of the pathogen associated nucleic acid sequences should decrease or become undetectable. With clinical relapse, the opposite should occur. Okay, so now what happened? Suppose a person get infected by the microorganisms, okay, and they are getting sick. So in the beginning, the number of the pathogen will be low. So the more sick the person will be, the number of the microbes will also increase. So number of the microbes means more nucleic acid. Less microorganisms, less nucleic acid. More microorganisms, more nucleic acid. So that means a very sick person will contain much more nucleic acid than of the candid of the pathogen than the less sick person. Okay. So now, if a sick person, okay, very sick person, he has lots of pathogenic nucleic acid. So lots of nucleic acid of the pathogen. So if he begin to get better, that means with the resolution of disease, he is getting better. So he will get better. At the same time, the number of nucleic acid of that pathogen would also get fewer. Okay, he will get better. He is getting less sick, so number of nucleic acid of the pathogen will also decrease. Okay, there should be a positive correlation. And in the contrary, if that person begin to get sick, the number of pathogen associated nucleic acid will also increase. There will be always a positive correlation. Okay, then another modification. When sequence detection predates disease or sequence copy number correlates with severity of the disease or pathology, the sequence disease association is more likely to be a causal relationship. This means, like, one thing I already described, this part, that sequence copy number correlates with severity of the disease. More sick, more nucleic acid of the pathogen. Less sick, less nucleic acid of the pathogen. Here, okay, so we understand that, but what is meant by it? When sequence detection predates disease, what it's mean? It's mean that, say for example, last year, this is 2020, so last year, say in 2019, some people get sick. Okay, suppose some people get sick in Dhaka, and that time some scientists, they isolated a microorganisms from that diseased person, and they analyzed genomes of that one. We can actually analyze the genomes by doing DNA sequencing. Okay, so in 2019, in 2019, some people get sick in Dhaka city, and some scientists isolated and sequenced some candidate pathogen from them. Okay, so now in 2020, again, some people, they get sick and the symptoms are similar like the disease that happened in 2019. Okay, please try to understand the scenario. So in 2020, again some people get sick and the symptoms relates with the previous disease, and again scientists isolated the isolate some pathogens and sequence them from these diseased people, and they find out that this sequence is similar with the sequence which was isolated or invented last year. Okay, so there is a chance that there is a positive relation, right? The disease was similar, and the isolated pathogens, that is the sequence of the isolated pathogens, are also similar. So it means there is a high chance that this particular disease is actually is caused by this particular microorganisms. Okay? Okay. Another modification. The nature of the microorganism inferred from the available sequence should be consistent with the known biological characteristics of that group of organisms. Okay, what that means? Let me explain with example of coronavirus. So I mean, now we know that the COVID-19, which is a coronavirus, which is causing the disease. So what happens in the beginning? People begin to get sick. So at the first, what people do, based on the symptoms of the infected person, the scientists, they try to guess what kind of, what can be the candidate pathogens. Okay, so now when in case of coronavirus, this recent outbreak, the COVID-19, so people get sick and based on the symptoms, okay, and those symptoms actually relates with some previous coronavirus outbreak. Okay, such as in 2004 there was a SARS outbreak, SARC. Okay, SARS. The SARS outbreak was also caused by another type of coronavirus. Okay, and in maybe around in 2009 or 10 or maybe in 12, I forgot the exact date. Around that time, another coronavirus outbreak happened in the Middle East, which is called MERS. Okay, so now when in 2000, in the beginning of the 2020, people people begin to get sick, and the symptoms are kind of similar with the previous coronavirus outbreaks. Okay, and then people isolated the people, they studied the sequence of of the causative agents, and they found that this sequence of these causative agents, they are similar with the previous outbreaks. Okay, so if this happens, if you find this one, then there is a high chance, okay, that this new disease is actually is caused by a similar type of pathogens which caused a similar type of disease in some previous date. Okay, okay, another modification. Tissue sequence correlates should be sought at the cellular level. Effort should be made to demonstrate a specific in situ hybridization or microbial sequence to areas of tissue pathology and to visible microorganisms or to areas where microorganisms are presumed to be located. Okay, so what that means? Suppose, say, coronavirus, it can affect our respiratory tract. Okay, it can affect our lung. Okay, so you want to say that coronavirus caused disease in our lung. So what you need to do, you have to prove that the coronavirus is present in my lung. Okay, this is actually important because say you some people get infected by the coronavirus and you take some sample from the throat. So if you take some sample from that throat, you actually can detect coronavirus and you can sequence them. Okay, but what you want to say, you want to say that coronavirus infect our lung. So you have to prove that the coronavirus sequence is present in the lung. So actually how we can prove that? There are different experimental methods. Okay, one method is you can take tissue from your lung. Okay, you have to take tissue from the infected portion of your lung, and then in those tissue you can detect the nucleic acid. Okay, you actually can do that. There is one technique which is called in situ hybridization. Okay, so right now just remember this name, in situ hybridization. Eventually you will learn more about these techniques, but right now just remember this thing. You have to prove that that microorganism is present in the diseased part or diseased tissue of your body. Okay. Okay, and finally the last modification. These sequence-based forms of evidence for microbial causation should be reproducible. This is very important in science. Your data should be reproducible. So, for example, with experiments, you proved all this statement, all these postulates for one person. When you will study with other person or in other area of this world, the data should be same. Okay. Everyone should get the same result. That means the data should be reproducible. Okay, so that are the major modification of the Koch postulates. Actually, nowadays people don't use the original Koch postulate as it has lot of limitations. Rather, we use this modified version of the Koch postulates. Okay, so that's all for this lecture. Thank you. See you in the next lecture. Till then, be safe. Thank you.